Well, fortunately or unfortunately, I'm the next speaker in the session. So perhaps I can request my colleague, Professor Varsha, to keep time, and then let me know when I cross. Yeah. Okay, so this work actually is, there are lots of contributors to this work, but I've only listed 3 names in the interest of space. Pun unintentional. Well, so basically what we've been trying to do is develop a payload, self-contained autonomous payload that can do monitoring of bacterial growth. So I'll give you a little bit of background information and then I'll tell you where we are. We've had 2 broad phases of development. The first phase, we had a prototype, and then now we're actually developing a model that should hopefully go on the first uncrewed Gaganyaan flight. Okay, so just to give some background, I'm not particularly well versed with biology, so if you will just pardon me some mistakes, this is a typical bioconsolidation process. The green ellipsoids are what are called urolytic bacteria, and they consolidate soil particles by releasing calcium carbonate from a calcium source. Okay, and the question that I think my friend colleague Alok has been working on, and his lab has been working on for a while, is to see if this process, which is very inherent to the Earth's soil and the Earth's lithosphere, can be used to make habitats in extraterrestrial space. So that's really the central question of all of this stuff. And I'll go through a quick timeline of what we've done and what we've shown so that it gives you some context. The first sort of step in the story is what we call space bricks, or what he calls space bricks. And they're made from this thing called lunar soil simulant. There are several soil simulants available. This is one that's provided by ISRO. And the process which we have sort of standardized is the following. So you have, this is like a typical casting process. I'm a manufacturing guy and this makes me very happy. And you have a mold which has a cavity inside, and then you have a slurry that has the bacterial solution with some nutrient and so on, along with the soil. And you put that into one of these cavities, one each, and then you ram it, and then you seal it with a paraffin strip, and then you let it set, right? And what you get at the end is a solid brick. And this is typical particle size in the lunar soil, that the LSS soil from ISRO. It's called ISAC-1, and you see that most of the soil particles are in sort of a 50 micron range. And at the end of this consolidation, you actually get something that has significant strength. So you see 2 axes here. This one gives you the maximum compressive stress that a sample can take. This is what the consolidated sample looks like. And this tells you roughly the toughness, so how long or how much energy the brick can take before it fails. And we've tried to add additives. Guar gum is one additive that we've been looking at for enhancing the strength, and then glass fiber was one of the additives we had early on for enhancing the toughness. And it seems to work reasonably well. The results are quite reproducible and so on. And the most surprising thing was you can actually take this brick and machine it. Right? You can actually cut it and it doesn't disintegrate. So it's actually fairly hard brick. Now, these bricks can also fail, and we've also investigated some of these failure mechanisms. It turns out porosity plays a central role in all of this stuff, right? And we've been developing some theory and numerics for trying to understand how this happens, and we've been trying to reproduce failure patterns and see what that tells us about the brick structure, about the porosity and things like that. The main thing is that if the brick is extremely porous, as the porosity, for example, the percentage of porosity goes up, this strength just falls very steeply, right? And it doesn't matter how strong the bond is between the soil particles that the bacteria makes. If the porosity is high, then inevitably it's going to fail, right? And that's what we found from the models and so on. And then sort of we took this from just the lunar soil to Martian soil, to go from Moon to Mars, so to speak. And this is the lunar soil solution, the lunar soil consolidation that I showed you earlier. And we can now make, or Alok's lab can make most of this from Martian soil as well. So this is a simulant that's obtained from the University of Central Florida called Exolith Labs. And the same bacteria with some tweaks can consolidate Martian soil as well. And the strength is also comparable to the lunar soil. It can actually take a few MPa of strength. I believe now we've gone up much more. To 5, even some samples have 10 MPa and so on. Okay, now the basic question is, this is all fine, but if you're talking about extraterrestrial habitats, so you have to be able to make this stuff in space, right? And so when we thought of this question, there are basically 2 steps. The first step, which is a little difficult, is to grow the bacteria in microgravity and see how they behave, right? So that's the first step, to characterize their activity and to see if there is any change because of microgravity, because of an extraterrestrial environment. And the second step, which is more difficult, is to evaluate the actual consolidation process. So you actually have a solution, you have the soil, and then you look at how it binds. That's a very difficult problem, as you can imagine, to have a self-contained payload to do that. So I'll only talk about this because there is not much that we've done. We're thinking about this still. So this is where this Manus payload comes in. It's short for microbial analysis in space, ANA's analysis. And we envisaged it at the start as a modular device. So there are 3 broad logic modules. There is what we call the cassette, which is where the experiment actually happens. And then there is something called a cartridge, which is a collection of cassettes. And then we have a housing which has the entire cartridge in it. And then we have a standard communication protocol, I2C, and so on. This is the first prototype that we built. This was published in 2021. Happened about a year ago. And you can see, for comparison, this is a 5 rupee coin, so it's about an inch and a half. And you see 3 cavities here. The little red spot you see is actually an LED. So what basically this entire thing does is to measure bacterial activity using optical density. Now, from a biology point of view, it's a very trivial experiment. But from a technology point of view, it's a little difficult to implement in a self-contained, remotely actuated package. So that's really where the challenge is. And basically, you have a little chamber where the bacteria is suspended in a solution. And then you have an LED light source at a particular wavelength. And then you pick up the intensity of the LED light with an optical sensor, right, a photodiode. And you monitor this as a function of time. So you keep querying the optical intensity, and you see how the intensity changes. And so from there, you infer how the bacteria is growing and things like that. And there's a simple sequence of things you have to do. So remember, this bacteria has to start growing when you want it to start growing, right? So you have to isolate, it turns out, the bacteria from its nutrient, because otherwise it will just start growing right away. And so we have a solenoid, we have a valve system. We had a valve system, we have a slightly better system now. That basically causes the 2 liquids to mix, the bacteria without the nutrient and the nutrient itself. And then once they mix, the bacteria comes in contact, the growth starts. So that is T equal to 0. And then we just periodically run the query, start the LED, wait for a couple of seconds, take a reading, stop. Start, take reading, stop. That's it, right? It's a very simple, conceptually simple experiment. So we did some experiments on Earth to simulate what we would encounter in microgravity. For instance, we made sure that the self-contained unit could reproduce or give us the same reading, unchanged reading, whether we were at low temperature, high temperature, low pressure, high pressure, and so on. Again, these pressures are not particularly small. 400 millibar is reasonably large compared to what you would see in outer space, but still, at least as a proof of concept, it tells us that it works. And we also did some very simple orientation tests, so you could tilt it in 2 different angles, and then you'll get the same reading without any change, right? So some of these standard tests we did, and we were able to make sure that the idea at least is operational. Okay, now we have something that we call Manus Mark II. That's a code word. We haven't quite named it yet. And it looks like this. So this is our final payload. This is the one that hopefully will be going on the first uncrewed Gaganyaan flight. It has 6 levels. And the first one is, the top and the bottom are basically the interface with the satellite on board which it sits. This one is a communication port, so it's a standard. If you've seen a computer VGA port, it looks something similar to that. It's called a D-sub. And the same thing happens here. The advantage is we can now control what each of these pins does, and then we have a PCB that breaks it out, and then there's a buck converter to step down the voltage and things like that. So the first level, I'll show you a breakup of the levels very quickly. The first level has basically all the power electronics shoved in. The second level has the flow control, so it has the system for controlling the motors and so on. And the third level has the actual pump, which will pump the liquid up from 2 reservoirs. One reservoir is the bacterial reservoir, one is the nutrient reservoir. And then there are valves, small micro valves, which will control the actual flow rate, and then you keep them closed before you start the experiment. And so that's how we're able to ensure that T equal to 0 is set when we want it to be set. And then there is the experimental chamber. So that little thing I showed you with the 5 rupee coin in front, that's actually here. A modified version of that is here. And then there is a data handling control unit on top, which collects the data and stores it on board and also transmits it to a communication system. This is the actual experimental module. So it's sort of blown up here. So this metallic piece has these inlets on all sides. That's where the fluid comes in. So there is one inlet, one outlet for each of these chambers. So you can see there are 4 chambers here. So there are 4 experiments. 1 is a control and 3 have bacteria in them. That's the plan. And there are O-rings here which are used to seed these on an acrylic plate. So it's optically transparent. So this is the LED source that you can measure. And the same thing is shown in the side view over here. You can see that the optical path length is 12 mm, which is more than the 10 mm that you need to get a valid signal. This is a blow up of the entire assembly. Again, it's just showing you each of these levels in the flesh. So the motor is here, the reservoir is sitting on the side. You can see the 2 of them here, and then the valves and the actual chamber and so on. We've also been doing some analysis. I'll spare you the details because they're not very interesting. It just says that this is good enough, right? So we've done modal frequency, we've done the stress strain, random vibration tests, harmonic tests, and so on. And I'll just end with this. So I would think of being right now at t equal to 0, which is the engineering, what ISRO calls the engineering model. And we're looking at mill-grade components in about 6 months. Hopefully we'll have a flight model by then. And demonstration of step 1 in 9 months. We're hoping that the flight will go in 9 months and we'll be able to demonstrate it at that point. And then design and testing for the second actual biosedimentation step is something that we're planning. It's a little ambitious, but we're hopeful we can pull it off. So yeah, so funding from both ISC, we were grateful enough to get a startup grant from ISC through the IOE program and ISRO also. So thank you so much.
Questions for Koushik? Okay.
So in those space bricks, the microbes are just one microbe, or can it also harbor biofilm communities?
I'm not sure if I'm qualified enough to answer that question. Maybe Alok or Varsha. Varsha is another collaborator, so maybe one of you guys can.
So right now it's just a single species chamber. So we can. It's not for biofilms yet. Okay, so
no, I think you're talking about the brick.
No. I got the.
Okay.
Hello?
Yeah.
So I had a question regarding the freezing and thawing cycle. So as you said, you guys are making the bricks type. Okay, so how you guys tackling the freezing and thawing? Because I heard that breaks over the time because of the freezing and thawing cycle due to the moisture.
You're talking about microgravity or the bricks?
Yeah, the brick stuff.
I'm not sure I
it
follow
was in
the
the first 2-3 slides.
I'll just quickly go to this. I'm not sure I follow the question though. Freezing and thawing in what? So you're talking about this?
Yes.
This, huh?
Yes. So due to the freezing and thawing, I have read that the bricks get weak over the time.
I don't think we encounter. Yeah, here it's just a room temperature casting experiment.
Oh, okay.
So it's just the only issue, well, slightly more mundane issue is stuff will get stuck to the wall. So you have something that is a barrier that allows you to take it off.
Okay.
So that's the only problem. Yeah.
Okay.
Yeah.
So I was impressed by those designs, how flexible they are. I mean, if they are to be used for some other application, can you tweak a little bit with them and adapt them to some other kind of applications?
Yeah, excellent. That's actually why we started on this whole modular thing, so that you don't want to go through the entire affair of doing the whole design again. So if, let's say, you change over to another bacteria or another species where only the actual measurement detail is changed, so then you have to change only the logic level at the cassette level, right? You don't have to mess with the other stuff for the most part. So that is sort of central to our entire design. So we hope that we'll be able to do. That's the plan, at least for us.
I'll talk to you more about this later.
Sure. Yes.
Okay. If there are no more questions, let's thank Koushik. Catch him at tea. Catch him at tea time.